Lesson 2 in animal responses is going to be covering the topic of nervous transmission. We're going to be looking at the generation and transmission of the nerve impulse in mammals to include the resting potential and how that is established and maintained, and how an action potential is generated. And we'll also be looking at how myelination affects this. One of the most important questions that we need to consider is what we understand by an electrical impulse. Now, in physics, of course, we know that electricity is the flow of charge and usually carried by electrons, but that won't be the same in the nerves. So we don't see electrons just moving along a nerve in the same way as we would along a wire. In fact, what's happening is that we're having the movement of ions, and the ions themselves are passing across the cell surface membrane, and that is creating a movement of electrical charge. So if we look at this simple diagram here of a motor neuron, we can see we've got two really important ions that we're interested in here. We've got potassium ions and we've got sodium ions, and it's their relative positions inside and outside of the axon which determines whether or not they are resting or whether they're actually allowing an impulse to pass along. So let's start with understanding resting potential. When a neuron is not transmitting an impulse, we described it as being in a resting state, and during that time there will be a resting potential which has been established. Now, this is created by a difference in charge between the inside and the outside of the axon. So when we talk about a resting potential, what we're really saying is that when the nerve is resting, there is a voltage or a potential difference across that membrane. So if we look at this axon here, this is the longitudinal section of the axon, and what we can see is we've got the two solid lines represent the cell surface membrane of the axon, and on the outside we've got a lot of positive charge, and on the inside we've got rows of negative charge. Now, this is a slightly deceptive kind of diagram because it suggests that we've got positive ions on the outside and negative ions on the inside. This diagram just showing you what the cross section of an axon would look like, with positive charge accumulating around the outside and negative charge on the inside. That is a total oversimplification of the situation, as you'll see as we go further on. But it does tell you the basic idea that we will relatively have more positive ions outside than inside. Now, in fact, actually what we've got is we've got positive ions on the outside and fewer positive ions on the inside. So we would describe the inside as being negative relative to the outside. So the membrane is then said to be polarized, and that then creates a potential difference and a charge. So if we have a look at this diagram, what we can see is we've got a microelectrode, and that microelectrode has been inserted into the membrane, and it's able to read the potential difference or the voltage that is created across that membrane. And this voltage, as I said earlier, is referred to as a potential difference. And when the cell is resting, then it's going to the voltage or the potential difference that's created is known as its resting potential. And in a resting axon, that would normally be about minus 70 millivolts, which means that it's really measuring the charge on the inside. So minus 70 millivolts tells me that it's negatively charged on the inside, and the potential difference is about 70. We'll start with how the neuron creates and maintains a resting potential. You have this diagram, so it'd be a good idea to label it at this point. We're just going to run through the structures you can see here. Starting off, we've got the phospholipid bilayer. Now, every membrane is created from a phospholipid bilayer, as you know, and wedged in amongst the phospholipids are a range of intrinsic proteins which span the entire width of the membrane. Now, this is the membrane which forms a major part of the axon. So obviously the axon has a membrane that completely surrounds the cytoplasm in the middle, and in amongst this axon you will have a range of different proteins. Firstly, we've got sodium-potassium pump, and this is a protein whose job is to move sodium and potassium across the membrane. Then we also have the sodium ion channels, another transport protein which can just allow the movement of sodium ions. And then we've got the potassium ion channels, which only allow the movement of potassium ions. So starting off, the resting potential is maintained first of all by the sodium-potassium pump, and the potassium channels will also play a part in this. So to start with, what's going to happen is we're going to be moving sodium and potassium ions. So sodium-potassium pumps will move 3 sodium ions out and 2 potassium ions in. So immediately we've got an imbalance in the quantity of charge on either side of the membrane. So we've got a greater quantity of positive charge on the outside compared to the inside. The sodium ion channels are locked, and these are closed at this point, and they will only be able to open when there's a change in the voltage across the membrane. So these ones will stop the sodium ions leaking back down the diffusion gradient. And this then maintains the quantity of sodium ions outside the axon. And this is then what we call the electrochemical gradient, and this is the sodium ion electrochemical gradient. So we've effectively got a high concentration of sodium on the outside and a low concentration of sodium ions on the inside. Now if we have a quick look at the potassium ion channels, what we find is that some of the potassium ion channels are open, and they're open quite a lot. So even though we've been pumping potassium ions into the axon, these potassium ions can travel back out down the diffusion gradient to the outside. So not all of the potassium ion channels are open. So we've got the one there which is circled in red, and that one is open, and the other one is closed. So a small amount of potassium ions drift back out from the interior to the exterior, which actually then also manages to increase that positive charge on the outside relative to the inside. So we will have a high concentration of sodium outside the axon, and there is a high concentration of potassium inside, but relatively there's a lot more sodium outside than inside, so we will end up with a difference in charge. So this then creates the potential difference across that membrane. Now when we look at the diagram, we tend to see the positive ions on the outside, and relatively we see that there is a negative charge on the inside. But I think, as I mentioned earlier, you've got to be careful, because it's not that the potassium ions carry a negative charge, they're just so much fewer of them that you will overall, this is more negative compared to the outside. So more positive charged potassium and sodium are now outside than inside, and this makes the inside negatively charged compared to the outside, and that will then measure a resting potential of minus 70 millivolts. You need to become familiar with this graph, because what it shows is it shows how the potential difference or voltage changes at any one point in an axon. So if you put a microelectrode into an axon at any position, then what we would see is we would see the voltage changing and following this pattern. So the first thing we've been talking about is resting potential. So on our graph, that would be this area of the graph, and you can see that when the neuron is resting, it's at minus 70 millivolts. So what we're going to look at next is what happens when the neurons are in fact sending an action potential, which is what happens when they have been stimulated. And this leads to what we call depolarization. There are a set sequence of events that are going to occur in the axon to pass the impulse along. So if we have a look at this diagram, we can just remind ourselves about the structure of the axon. Over here we've got a potassium ion channel that can be opened and closed. Over here we've got another potassium ion channel that's open all the time. So this is the ion channel, which really allows the diffusion of potassium ions out of the axon down the concentration gradient, regardless of how much potassium is being pumped in by the sodium potassium pump. And in the middle here we have some sodium ion channels, and these ones are closed when the conditions of a certain type of voltage will keep them closed. So they need a certain change in potential difference or voltage in order for them to open. So what we can see on this slide is the diagram I've just described to you, where you've got the phospholipid bilayer and you've got the various different channel proteins which are embedded in the phospholipid bilayer. Some of them are open and some of them are closed. This diagram is showing the various different positions of charge on a membrane. So we've got outside the axon and we've got inside the axon. So, and in between, in this sort of pink, you've got the phospholipid bilayer, the membrane, which divides the two. Now you can see we've got where we have a resting potential, where we've got positive charge on the outside and negative charge relatively on the inside. And then we've got something called the action potential, which is what we're going to be discussing in more detail. And you can see there's a reversal of the charge. So when a neuron is stimulated, sodium ions channels in the cell membrane are going to be able to open. And so this allows the sodium ions to start to flood into the axon down the concentration gradient. And this is going to cause a rapid change in the potential difference at that point in the membrane. Now obviously not the whole length of the membrane, but only at that particular point. So if we look back to our graph that we talked about earlier, what we would see is we would see the stimulus happening at this point and the sodium channels open. Now the stimulus that's needed to actually open these channels will vary depending upon what the receptor is. So for example, it could be a simple physical pressure that opens the sodium channels, or it could be the energy from light, depending upon what type of receptor we're talking about. But whatever the stimulus is, it will allow the sodium ions to diffuse into the axon. That's following the electrochemical gradient, and that will then turn the potential difference from minus 70 millivolts to something going to 0. So on our graph, we're going to be in that section of the graph. The diagram on the left there shows what the axon looks like when it's resting. We've got the positive charge on the outside and the negative charge on the inside. And at the top right, we can see the diagram where sodium ions are now able to access the inside of the axon, and that's because the voltage gated sodium channels have been opened due to the stimulus. This is known as depolarization. So at that particular point in the membrane, that particular area has become depolarized. Now if enough sodium ions manage to enter into the axon, then the potential difference will start moving from minus 70 millivolts towards minus 55 millivolts. And this is what we call the threshold value. When enough sodium ions have entered the axon and the voltage across the membrane has gone down to, say for example, let's say it's gone to minus 50 millivolts, that's exceeded the threshold value, and that will cause even more of the voltage gated sodium channels to open. So all of the other ones in the nearby area will open up. And this will then cause more sodium ions to diffuse into the axon. Now, this is an example of positive feedback. We've obviously come across negative feedback where a change results in a response which reverses that change. Now positive feedback is where a change has happened and the response is to make it happen even more. So you're actually increasing the effect. Now if we look at this diagram, we can see that we've got the axon now, and at that very left hand side we've got a reversal of charge because the sodium ions have now moved into the axon. And so now we've got positive ions on the inside of the axon and negatively charged ions relatively on the outside. And so that point in the axon has been depolarized. Now in the axon we've got a lot of these voltage gated sodium channels, and when a small number of sodiums enter into the axon, it causes a change in voltage, and it actually causes the rest of the sodium channels shape to change, and that then allows even more sodium ions to enter. Now, I've just mentioned that this is a process called positive feedback, and this amplifies the change away from the normal level. So we've got the resting potential, which causes the sodium channels to open. The sodium then diffuse in. This causes depolarization, and then we've got the voltage gated sodium channels opening, and that then goes back to the sodium channels opening and even more sodium diffusing in. So we're actually increasing the effect. Now, if we look on this diagram again, the area of the graph that we're now referring to is just here. So what we can see now, we've exceeded the threshold. All of the voltage gated sodium channels are opening in that area, and as much sodium as possible is diffusing into the axon following the concentration gradient. And this will then cause, at that particular point, a huge reversal of the charge, so that you go from where we started at minus 70 millivolts, and you're going to get up to about 35 to 40 millivolts. So this can be just shown with this simple diagram. So sodium channels are open and sodium ions diffusing in, causing depolarization at that point in the axon. So minus 70 millivolts changes to about plus 30 millivolts. Now we get to this point when the as much sodium as is possible has now got in, and then this causes the sodium channels to close, and the next thing that happens is the potassium channels open. Some of them, of course, were already open, but now we're going to be looking at the ones that were closed until this point. So we have this stage now called repolarization, and this is what happens. Once we get to about 30 millivolts, the sodium ion channels are going to close, and the voltage gated potassium ions are going to open. So if we look here, we can see that these potassium ion channels are now opening. And this means then that the positive charge inside the axon is now going to start moving out again. So we're going to see a fall in the positive charge inside the axon, so as the sodium starts to diffuse out. So sodium starts to diffuse out the neuron following the electrochemical gradient, and this will then move the membrane back towards its original resting potential. So we are going to start becoming more negative on the inside. So looking back at this diagram, if we look at the where we started, we've started now going back to having positive charge on the outside and negative charge on the inside. And what you can see is, you can also see that the depolarization has actually moved forward in the axon. So as the charge reaches 30 millivolts, the sodium channels close, the voltage gated potassium channels open, allowing the potassium to diffuse out, reducing the potential difference back towards a negative value. The axon becomes negatively charged inside, and the action potential moves forward as the next section is depolarized. We've now reached this stage where the potential difference has dropped down to less than where we started. This is known as hyperpolarization. Very simply, all that happens is, as the potassium ions have been diffusing out of the axon, some of the voltage gated channels take a little bit longer to close, so more of the potassium ions diffuse out than you would perhaps expect, and that then creates a more negative value of the charge on the inside. We can see that here on this diagram. So we would started off at about minus 70, but we've actually gone to perhaps minus 85, something like that. So this stage is known as hyperpolarization, and eventually the voltage gated potassium channels will close, and some of, as we've said before, some of the potassium channels stay open because they're not controlled by voltage. Eventually the resting potential will be restored, and this is because the sodium potassium pumps now have to return the ions to their original starting points. So at that point in the graph we've actually managed to return the sodium ions back outside and the potassium ions back inside. So if you remember, we've got 3 sodium ions being pumped out and 2 potassium ions being pumped in, and this will then restore the membrane to its resting potential. And until that happens, the nerve isn't going to be in a position to actually send any more impulses. And it's also worth remembering that because we've got these potassium ion channels open, some of them open, that even when the sodium potassium pump is moving these ions to their correct positions, the potassium ions are still able to diffuse back out a bit, which is why you still have this difference in the charge. Now we've entered this stage called the refractory period, and you can see that, which is bracketed on that diagram, and this is where the nerve is unable to send another action potential, and this is because the ion channels aren't able to open again at this stage. And this means as well that the sodium ions particularly won't be open because we haven't repolarized the membrane. So basically it's like having dominoes all stacked up to sort of knock them over, and unless they're all stacked up again you can't knock them over again. And so that is the way the refractory period is working. Now this brings us on to this idea of propagation of an action potential. The idea that we can make this nerve impulse travel from one end of the axon to the other. So how does the way that the ions move ensure that the action potential is going to move along the axon? Now the first thing that happens in an action potential is that the sodium ions diffuse into the axon. Some of them will diffuse slightly sideways, and what that does is the charge on those sodium ions will affect the slightly more negative charge in the other ions around it, and that then stimulates that membrane so that the sodium channels will open. And as those ones, the sodium channels open, those sodium ions diffuse in, and they also diffuse sideways as well and affect the next ones, the next sodium channels that are nearby. And in that way the action potential is moved along the axon. We have this thing, this diagram shows the action potential moving along in the opposite direction from the right to the left, and what you need to do is also notice you've got the red line there which shows the resting potential and the action potential, and it's the line that you've got on that graph that we did earlier, and you'll see that ultimately what's happening is that peak of the action potential is just travelling along like a wave along the axon. So the resting potential is caused when we've got the concentration of sodium ions high on the outside relative to the inside, and the potassium is high on the inside compared to the outside, but we still have an overall positive charge on the outside. The stimulus causes the sodium ions to move in, and this is generating the action potential. Now at the point where the action potential is happening, the membrane is depolarized. So if we look at the diagram we've got there, we've got the blue area which is the polarized, and then we've got sort of orangey red area which is the depolarized. Some of the sodium ions then are going to diffuse sideways, and we've also got some negative charge, which means we've got some actual negatively charged ions inside the axon, which we haven't talked about very much, but they are there. if you have a look on this diagram, you can see that you've got this kind of circulating charge and around the membrane. So we've got positive and negative, and this actually means that charge is effectively circulating in this border region between the blue polarized area and the red depolarized area, and these are referred to as localized currents. They cause the voltage gated sodium channels in the next region to open, which causes an influx of these sodium ions, and then we get a new area that is depolarized. So behind the area of depolarization, the sodium voltage gated channels are going to close, and this is then going to allow the potassium voltage gated channels to open, and so the potassium is now going to diffuse out of the axon and into the area around the axon, and this then leads to repolarization and hyperpolarization, which is shown in yellow. So what we can see is we've got these three regions in the axon. To start with, we've got a blue polarized region, then we've got the sort of orangey red depolarized region, and then we've got the yellow of the repolarized region. And these three phases are going to be moving down the axon. After the repolarization and hyperpolarization, the membrane is going to then be returned to its original state, and it's going to go back into its resting potential phase, and that's going to happen as the sodium potassium pump restores the sodium to the outside and the potassium to the inside. Now, of course, we've also had this refractory period as well, which occurred during the repolarization and hyperpolarization stage. This is all put together and added to this diagram. It's not very clear because the colours haven't come out particularly well on here, but we can see quite clearly the polarised regions in blue. So these are either ends of this peak. We've got the area where there's the main peak of the depolarization, and that's the sort of orangey bit, and then you've got this repolarization and hyperpolarization stage, which is the yellowy bit, and you can see the impulse is moving from the right side of the page to the left side of the page. I'll give you the link for this video, which shows an action potential travelling down. But we can add to this that therefore the propagation of the action potential is always in one direction along the neurone. And again, we can think of it like dominoes toppling, or like a Mexican wave. Sodium ions rushing in, creating a wave of activity. And what you're seeing is that I want you to think about the fact that the ions themselves are not actually moving along the axon. All that's happening is that the ions are actually moving in and out of the axon at fixed points. It's like a person standing up and sitting down. They haven't moved around the stadium with the wave. They've stayed still. And actually the sodium and the potassium ions aren't travelling down the axon. What they're actually doing is they're just moving in and out of the axon at points nearby. And that movement of the sodium and the potassium is travelling along the axon, but not the sodium and the potassium themselves. I want to just remind ourselves about the importance of the refractory period. So after an action potential, the neuron cannot be excited again straight away. And that's because the ion channels that have allowed the ions in and out are still recovering and won't open again. So during the repolarization stage, the sodium channels will be closed, and during this time that the sodium channels can't be opened, this is known as the refractory period, and we can see that at this point on this graph. So it's around about the time of hyperpolarization and at the end of the depolarization. So it makes sense to have some idea as to why a refractory period has evolved. Now, there is an important value to the refractory period. There's a time delay between one action potential and the next, so it means that there is going to be a very distinct gap between one impulse and the next one that travels down, and this makes sure that action potentials are not going to overlap and will pass down as separate impulses. And it also means, most importantly, that the action potentials have to move in one direction, because as soon as one area of the nerve has been activated, the bit behind it can no longer allow the action potential. So the only place that the action potential can move is forward, because it can't move backwards, because that area has now effectively closed down while it's in the refractory period. I will just say that if you look on the diagram, you can see that refractory period is a very short period of time. It's only about 1 millisecond, so it's not a huge amount of time, but it's long enough to prevent the ions diffusing backwards and therefore the impulses travelling in a much more random fashion, which obviously would be of no value whatsoever. This is referred to as unidirectional wave of depolarization. So the wave depolarization cannot move back towards the areas which are in a refractory period, because they're not able to have an action potential because the sodium channels are closed, and this means then that the action potential can only move forward where the voltage gated channels are able to be opened. This now brings us on to something called saltatory conduction. We've mentioned myelinated neurons before, and myelinated neurons are ones which have this type of insulation caused by these Schwann cells, and they will allow the impulses to travel much faster than non-myelinated neurons. And this is because the depolarization that we've talked about can only occur at the nodes of Ranvier. So this is only at these places where you don't have this insulation can the sodium and potassium ions move in and out of the axon. So if we look at this diagram, what we can see that the sodium ions are being able to diffuse out at point B, and then the impulse has to carry on right down to point C. And so the impulse is literally leaping from one node to the next. Now this means, of course, that the process is going to be a lot faster than if it was going to have to travel through all of the different channels all the way along the axon. So saltatory conduction is much faster than the wave. Every time a channel opens, the ions will take time to diffuse in and out. So if you only have to have that movement at particular points along the axon, it's going to be much faster than if you had to wait for every single one to carry this process out. So we're going to have a much faster transmission. Another value of the myelin sheath is it manages to keep the ions in their right position, so you get less this sort of drifting of the sodium and potassium ions through the membrane because the myelin is there. And also the cations might move towards the anions along the inner surface of the membrane, and those that do are effectively neutralised, so they don't actually create the charge that you would want. So it actually allows this the charge to become more stable. The other value of saltatory conduction, not only is it very fast, but it's also very much more energy efficient. Wherever you have the movement of sodium and potassium ions using the protein pumps, it's going to require quite a lot of ATP to move them against their concentration gradients, because this of course is active transport. So of course that requires energy. So if you only have to set up a resting potential in the positions of the nodes of Ranvier, then it's going to mean you're going to need to use less energy to do that. And you also need less repolarization with the myelinated neurons. This brings us on to factors affecting the impulse speed. The axon diameter. It's very simply, the greater the diameter of the axon, the faster the impulse is going to pass. And that's very simply because the bigger the diameter, the more sodium and potassium carrier proteins you're going to have in the membrane, and therefore you're going to have greater places for the ions to flow in and out. And you can see that here on this diagram with the graph. You can see as the diameter increases, the conductivity and the speed increases. Temperature. When an organism is warmer, then the impulse is going to be faster, and that's very simply because ions diffuse quicker at higher temperatures. I think it's always tempting to think of temperature in terms of enzymes, but in this case, because we're talking about diffusion of ions, it's worth remembering that ions diffuse faster when they're warmer because they have more kinetic energy. This brings us on to the all or nothing principle. Nerve impulses are said to be all or nothing responses. What this means is that every action potential that travels along a neuron has the same size, and as long as it achieves the threshold value, you will get an action potential. And if you haven't achieved that threshold value, then there isn't going to be an action potential and you will have no response at all. So no matter how big a stimulus is, you will still get the same size action potential, which may sound very strange because you'll think sometimes if you're touched, you can notice that the touch is very gentle and light, or you could be touched in a very sort of harsh way, so you know that actually it does feel different. And that isn't to do with the size of the action potential, but it's to do with the frequency of the action potential. So this information is summarised here with these graphs. The size of the action potential isn't changed, but the number of stimuli is. So we've got the top there, a weak stimulus, and you can only see we've got one action potential. And then as you go down towards the bottom, we've got a very strong stimulus with lots of action potentials. So the larger the stimulus, the more frequently the action potentials are generated. And that allows the body to have a maximum number of stimuli that it can receive as well. And also the other thing that is beneficial for this is because we have a same size action potential every time, this all or nothing idea means that if you only have a very, very fine stimulus, it might not be enough to actually generate an action potential at all. Now that brings us to the end of the theory for the way that nerve impulses are transmitted, and the rest of this presentation gives you a range of questions. And I'm going to allow you to have a look at these questions, which then follow on with some answers, but I'm not going to talk through it. I'm just going to allow you to have a look at it, and you can use your own time to go through and then see how much of this information you can recall by looking at the answers that go with it. Thank you very much, everybody, and I hope you have found this useful.